we will go to blast. I think many of people are left, many are there. I will go to blast. So we are doing a lot of things today, please be there. NCBA blast. So NCBA blast is a basic local alignment search tool which take our sequences and search it against database. I will just take a sequence for that, a protein sequence I will search. I have just taken a sequences for multiple sequence alignment here. I will just take a sequence from that and do the blast, and then we will see that. So blast can be of different types. Blast, what it is, if we give a sequence, it find out the similar sequences by searching throughout the database, and it will align the sequences. It will do pairwise alignment and find out the best sequences, best similar sequences. We can do a protein blast, nucleotide blast, or blast X, mainly used for translated nucleotide against protein. Actually, these blast X all we can do for the in silico translated nucleotide sequence. We can search against a protein database. Mainly we do for while predicting gene, all these things. Usually if we don't have a protein sequence of a particular nucleotide, we will go for the translation of the nucleotide, and then translated sequence can be searched against protein to confirm whether that protein is the identity of the protein. Or T-blast and protein can be compared with the translated nucleotide present in the database. So these are some of the most used blast. I will just go for protein blast. Just go for protein blast. As you all know, everyone, I think here have we already done this blast. We can give accession number, or you can say give a genome identifier number, that is the GI number or a FASTA sequence. I will just paste the FASTA sequence, and you can determine the query sub range. If you need to go do for the blast between first residue and 50th residue only, we can do that. Or you can upload the file using the upload, and you can give a job title if you need. It's an optional one, and you can just there is experimental database, this new one. We can search against standard nucleotide database or experimental database. Experimental custard nucleotide database, I have not gone through that. Standard database we are just searching against, and non-redundant protein against non-redundant protein sequence we are searching. Redundant means repetitive sequences. Sometimes when we submit a sequences multiple times, we can the error can occur, because in my case I have done a sequencing of bacteria. At the same time, one of my friend also have shared the sequences, but unknowingly she also submitted to NCBA. I also submitted to NCBA, and we have multiple entries of the same sequence in the NCBA. So that is called redundant sequence. So it should not be happen. So non-redundant sequence, we should compare the non-redundant sequence, and we can compare with the particular organism. If you need your sequence to compare with a particular organism only, you can give that. And non-redundant proteins reference sequences, if you need to compare, or you can exclude these things. If uncultured environmental samples they are comparing. Uncultured means environmental samples, metagenomic samples, some of the bacteria may not be found out. They will show unidentified bacteria, unidentified protein, like that. You can just opt out them. Or non-redundant RF6 protein, you can opt out. So like that you can opt out. And blast P we are doing, or we can select different types of other blast. So you are just clicking the blast. The blast program will go and find out the similar sequences. I think everyone has done this, and you people all know that what is blast. It will find out the and this was, as I said in the initial classes, this was found out by Alshul, and this was one of the very highly cited technology or a highly cited tool, which cited more than 1 lakh, and it is for the surprise it is published in a journal of impact factor of 5 or something, less than 5 I think. So it is one of the most widely used tool among the biologists, and we have just used the BLAST P. And what BLAST do is that BLAST usually break down when you give the sequences. BLAST usually break down the sequences into words. They are called k tuples, etc. So they will break down the sequence into number of words. They will first break down the sequence into this word, and then they will start from the here, and then they will break down a 4 number word again. Then they will start from the third, and then they will break down into another 4, and then they will start from this V, and they will break down the sequence into another 4. Like this, they will break down the sequence into this 4 words, 4 letter words. They will break down the sequence, and each word will be searched against a database sequence. If a word get a similarity between a database sequence, we will try to align the rest of the sequence in both the sides, and try to find out whether the sequence, we will try to align the rest of the sequence with this, and try to find out whether it is showing some similarity with the neighboring sequences. If they are showing the similarity at a particular threshold, we will proceed with that alignment. But if we, because they are converting it to small words, the small word will bind, and after that when we try to align the sequences, align that full sequences with that region, if there is no alignment or no scoring we are getting with their threshold they have already calculated, if we not getting a proper score, we will not go with that alignment, we will go with another alignment. So like that, they will try to align. We got a hit with a word only. After the hitting with that word, we will try to align the rest of the sequence with that database sequence we found out. And if we get a you can see, if we get a similarity with that, we will proceed with that alignment. If we don't get a similarity, we will proceed with some other alignment. This is how BLAST search. This is what BLAST is doing with our sequence now. It is doing, BLAST P is doing the search now. We will go and check the result. So BLAST will cut our sequence into words. It served as k tuples, all these things, and these words are searched against a database. If one word is showing a similarity with one of the sequence in the database, we will try to extend that word in both directions, extend that word rest of that sequence with in both direction at with the database sequence, and find out whether we can get a better score with that sequence. If we are getting better score, we will follow that alignment. This is what BLAST is doing. So in BLAST we can limit with by organism, as I said. We can give we can limit our search with either human DNA or E. coli DNA, or several limits can be done. I have already explained in that BLAST query, and several limits and searching we can do many things. And we will go for the BLAST result now. I think our BLAST result is ready now. So this is about BLAST, what a BLAST algorithm do. So this is the BLAST result. So we can see here our hexokinase is being aligned to the aligned, and the BLAST P result we can see this compared with the nucleated sequences and the query ID they have given, and the title of our sequence, we can give the title which will be shown, and the molecular type is amino acid, our length of the query, and what method is being used, and all these things we will be showing. And we can see here the a chart showing the similar sequences. The most similar sequences will be seen at first. It will be same as what we taken from the what I taken from the NCBA. So less similar sequences we can see down. So the most similar sequence will be seen at the first or the top of the this table, and we can see the maximum, the score here, and total score, maximum score, and then we can see the query cover, how much the query is covered. Cover means query cover means how much the query is been aligned with the how much the this is the query or the sequence which we have given, how much it is aligned with the database sequence. It is 100% database sequence was aligned. If the query is aligned with only 50% of the database sequence, we can the query the coverage become 50% only. So the query is aligned with the database sequence 100%. So we got the query coverage 100%, and then there is a e value is there, and identity is there, how much identity between the sequences, and there is 100% identity. So actually that sequence is taken from NCBA, and the sequence length and accession number all are there. And e value is the value which denotes the similarity between the sequence, or it's a statistical value which express the similarity between the sequence. And lower the e value which start from 0, and lower the e value, the better the alignment between. Most of this having the 0 e value, lower the e value, the better the alignment between the sequence is been. So we can see what is the things. So we have not gone through the graphical display, we will come there. So the list table, or list of its table which we have already seen now, that is a list of hit table. So the list of hit table tabulates a target sequence that produce the significant alignment. We have seen the alignment of our query sequence with the database sequence, along with the corresponding bit score is there. Earlier bit score was there, now the bit score is not there. And the query coverage, we have seen the query coverage, how much the query is being and the target database sequence, how much it is covered, how much of the database sequence is covered compared to the query. That is how much percentage the query is aligned with the database sequence. That is the query coverage we have already discussed. And e value is there, identity percentage is there, and assigned accession number is there. We already see. And the individual alignments we can see. We can see the individual alignments. I think we can see the individual alignments here. We can see the each pairwise alignment. We can see the query with the subject, the database sequence alignment we can see here. So how the query is being aligned with the subject, you can see identity is full identity because we already done that. Already we have taken the same sequence. So the alignment with the query can be seen here. So the alignment can be seen. So the individual alignment along with the calculated parameters can be seen. Individual alignment provides more details about the similarity between the sequences. E value, bit score, percent identity can be calculated for each alignment, and you will also find gaps and clusters of the identical sequence with the individual alignments in the panel. You can see the gaps or anything between the sequences. I think we are not seeing anything in our thing. And then the important thing is the e value. What is e value? E value, the percentage similarity between two or more sequence alone is not enough to ensure trustworthy alignment. So bioinformatics buff tend to regard e value as the most informative parameter looking for true matches. E value is a statistical value calculated by BLAST, which is used to tell about which gives the information about the true matches. To keep simple, e value take into account of number of hits one can expect to obtain by chance when searching in a given database. Represent the probability of a given sequence is a significant match. So, number of hits one can expect to obtain by chance, or the probability of a given sequence to show a significant match with the database sequence, that is the E value. So, lower the E value, more significant is the alignment. That is the probability of a query sequence to get a significant match with the database sequence, or we can say the number of hits one can expect to obtain by chance when searching a given database. It is simply saying probability of a given sequence to get a significant match or alignment between the database sequence. That is called, from that only the E value is being calculated. The lower the E value, the better the alignment is. 0 is the best E value, where the alignment, the E value is the best E value. So, E values of alignment alone may be useful tool for rapidly search different databases of query. Same thing is the same. So, E value and same things only. That's all. So, blast output, and we can see a graphical representation also. You see that graphical representation, and go to the cluster W, and there is a graphical summary is there. Different colors are used to show this is our query sequence, and you can see the query sequence here. Coverage is less, hexakinase drosophila. You can see the query is our query is big, and the sequence in the database is small. It is not fully covered. That is what here also the you can see the database sequence is long. Here database sequence is long, and our query sequence is not much covered. So, like that different color code they will give. You can see the color codes. You can see if the alignment score is greater than 200, the best alignment score greater than 200, the red color they will show. The query and the database sequence are of very good alignment score, and if the alignment score is 80 to 200, these lines will be of blue color, and if it is alignment score is less than 40, it will be black in color. These lines will be. So, this is our query sequence, and these are the sequence from the database. So, this is